Okay, well, welcome everyone to today's webinar hosted by the Bot-Stieber Institute for Wildlife Fertility Control. My name is Monique Principe, and I'm the Managing Director of the Institute. And today's webinar is titled Gene Delivery Approach with AMH for Single Dose Lifetime Contraception for Female Cats. We are very fortunate to welcome two presenters today. Philippe Godin and Lindsey Vansandt. And I'll just give a quick summary of their affiliations and background. Philippe Godin is a doctor of veterinary medicine and holds a PhD in veterinary sciences with a focus on ovarian physiology. And both those degrees were at the University of Montreal. His dissertation focused on the characterization of the cellular signaling during the ovulation process. He is currently appointed as a postdoctoral fellow at Massachusetts General Hospital, Harvard Medical School. And Lindsey Vansandt is director of the Imperiled Cat Signature Project at the Center for Conservation and Research of Endangered Wildlife at the Cincinnati Zoo and Botanical Garden. Lindsey is a graduate of the University of Missouri School of Veterinary Medicine. She obtained a PhD in animal sciences at the University of Maryland in collaboration with the Smithsonian Conservation Biology Institute. She sits on the Scientific Advisory Board of the Michelson Prize and Grants in Reproductive Biology Program and the Association of Zoos and Aquariums Reproductive Management Advisory Board. Also joining us today is Stephanie Boyles-Griffin. Stephanie, would you like to introduce yourself and say hello?
Hello, everybody. Stephanie Boyles-Griffin. I serve as the chair of the advisory committee for the Bot-Stieber Institute for Wildlife Fertility Control. I also served as a senior scientist and program director at the Humane Society of the United States Wildlife Protection Department.
Okay, so I welcome everyone, and let me just explain how the question and answer session will work following the presentations. You'll type in your questions into the Q&A box on your screen, and you can type those in at any time and they will accumulate for us. And Stephanie will moderate that session. She will read all the questions aloud, and then our presenters will answer them during the webinar. And if there are any questions that we don't get to, we can provide follow-up for you if you want to reach out to the Bot-Stieber Institute at biwfc@botstieber.org, and we can make sure those questions are answered. I don't think there's anything else. I think we are ready to proceed, so let's begin the presentation.
Great. I'm going to start things off, and then Lindsey will do the second half of the presentation. Are you able to see my slide? You can hear me well?
Yes, it all looks good.
Great. Okay, well, thanks, Monique. Thanks to the Institute for inviting Lindsey and I to present our new gene therapy approach to contraception and its application in female domestic cats. Uncontrolled reproduction of unowned free-roaming cats, it's an animal welfare concern. It's estimated that there's hundreds of millions of those free-roaming feral and stray cats worldwide. And these cats, well, they don't have access to proper veterinary care. So they are more likely to get infectious diseases. They get into fights. They're more likely to be involved in traffic accidents. And these factors, they all contribute to a reduced lifespan. And it's not only a welfare concern for the cats themselves, but they are also putting an immense predation pressure on small wildlife. I'm showing you this picture here. It's an award-winning picture of all the different animals that were brought to a veterinary clinic because they were injured by cats. And I show you this to show you how cats, they are generalist predators. They can prey on various species of small birds, mammals, and reptiles. And they are especially harmful to regions where animal did not evolve any defense mechanism against those very good hunters. So there's many strategies to manage these cat populations. Culling is not very sensible strategy to control those populations of animals that we consider companion animals. And it's not even scientifically proven to be effective. Since you remove animals, more cats will immigrate in this area, or there would be better survival rates for the next generation. Same thing goes for only translocating these animals. Adoption is a good strategy, but cannot solve the problem of those hundreds of millions of cats, and not all feral cats have the potential to live in an household environment. I mean, these cats are such prolific breeders. They can have their first litter well before 1 year of age, and they can raise multiple litters every year. And Lindsey will discuss more about this, but cats, they are induced ovulators. So it's one of the best strategies to make sure that you get often pregnant, because it's the physical act of mating that induces ovulation in that species. Another strategy to control the population is through the surgical sterilization of as many individuals as possible. And those campaigns are called TNR. So trap, neuter, return, neuter or spay. So the goal is to capture the animal, spay or neuter them, then release them back in the environment. This requires a lot of logistics. It's time and resource intensive because it requires trained personnel to perform the surgeries and specialized equipments. So this is good to control like local semi-closed populations, but it cannot scale to control the hundreds of millions of free-roaming cats in the world. In fact, the development of a non-surgical contraceptive would truly revolutionize the way we manage those populations. And immunocontraceptive vaccines have been tested in the domestic cats because it works in many other species. But unfortunately, the vaccine against Zona pellucida, yes, it raises antibodies in the animals, but those antibodies are not able to recognize the feline Zona pellucida, and the animals still can get pregnant. And the GnRH vaccine, well, it has a variable efficacy, and this efficacy is not sufficient to justify its like wide use for control of free-roaming cat populations. An ideal contraceptive would be a single injection that covers the whole reproductive lifespan of the animal, that would be affordable and easy to implement in the field. The Michelson Animal Foundation is providing grants to stimulate research in the area of finding this type of contraceptive for cats and dogs. And thanks to those grants, our team at MGH in the lab of Dr. David Pepin and our collaborators at the Center for Conservation and Research of Endangered Wildlife crew, we were able to unite and work on their finding this kind of contraceptive. So today we're not presenting a success story in the field, rather we're presenting the results of a proof of concept study that we published in June, where we showed that a gene therapy works in a laboratory environment, and you can access this paper online, but you can also, for a shorter read, see the media coverage we got after the publication of this paper. So my job here for this presentation is to introduce you to the gene therapy itself, AMH, so anti-Mullerian hormone, and kind of bring you an understanding of the biology behind all of this. And then Lindsey will present the exciting results we got with this therapy. So anti-Mullerian hormone, it's a natural hormone, it's a protein. It was first discovered as having a role in the development of the reproductive tract, like in utero, in fetuses, but it's also involved in the adult reproductive physiology. So in a female, AMH, the hormone, is produced by the granulosa cells. So here on the image, you can see it's an ovarian follicle, so it's the functional unit in the ovary. There's an oocyte in the middle that will be ovulated, and surrounding it, there's the granules of cells. So these are the cells that produce AMH. So for those of you that are less familiar with ovarian biology, those follicles, they are quite small at the primordial stage. So when they're very, very small, and then they're able to grow, become bigger, and eventually some of them, not all of them, will be able to ovulate and release the oocyte. And it's the secondary and small antral follicles that produce most of the AMH that is present in the blood of the female. AMH signaling is quite conserved in invertebrates. Its expression has been detected in all vertebrates in which it has been studied. So that is more than 20 species of fish, handful of reptiles and amphibians, and most mammals and birds that were studied. And in birds and mammals, AMH has been proven to be an ovarian inhibitor in these species. So its role in the adult cycling female, it's to slow down the primordial follicle activation. So the passage from this small stage of follicle to the later stages, it inhibits this, and also slows down the preantral follicle development. So the best way to understand its role is to make sure that not all follicles will grow, to make sure that we will still have a follicle reserve that will remain and not be depleted too fast, in order to make sure that you're able to ovulate eggs for the whole reproductive lifespan of any animal. And the best way to see this is looking at the results researcher got when looking at ovaries of mice that were lacking the AMH gene. So here in the darker bar, it's mice that lacks a normal allele for AMH. So they're lacking AMH. At 4 months of age, they have less primordial follicles in their ovaries, and this remains the same at 13 months of age. The same researchers, they took ovaries from 2 days old mice and they put them in culture. And some of them were treated with AMH, so the darker bar. And as you can see, there's less growing follicles when ovaries are treated with AMH. So it shows that it's a good inhibitor of ovarian growth. But this result is in vitro, but what would happen if we would sustain high AMH level in a whole organism? So that's exactly what our lab did. So you can see here, it's the ovaries of a control animal and one that was under high AMH level. So their ovaries are way smaller. The reason for this, so if you focus on the purple bar here, it's the number of follicles for each stages. So mice that were injected AMH twice daily for 40 days, they have significantly less secondary and antral follicles. So those bigger follicles that lead to ovulation, they almost have none of them. But these injections twice daily for 40 days, it's quite labor intensive, and it's really not applicable for a contraceptive strategy of feral animals. And that's where the gene therapy comes into play. So the red bar is gene therapy using AAVs, a single injection. 40 days later, we got the same effect than if we would to inject the AMH protein twice daily. So this gene therapy, it's using adeno-associated viruses, so AAVs. They are non-pathogenic, non-replicative, small viruses with very low immunogenicity. And that makes them ideal vectors. So their job is to deliver a transgene. And they are already used, they are approved by the FDA for 4 different gene therapies. And in these therapies, they are used to deliver an intact gene to human patients that are afflicted with rare genetic diseases. So you give the intact copy of a gene to people that have mutations. So to make it more visual, we're using the ability of the AAVs to internalize in cells and to deliver their genetic content in the nucleus of the cells. Then the cell is able to take the DNA template we provided in the AAV capsid, and it can produce unrestricted amounts of AMH that hopefully will be enough to inhibit ovarian activity. So that's the goal. So instead of giving the protein, we're giving the DNA template for the cell to produce it themselves. So the AAV-AMH combination of an AMH gene and AAVs is a dose-dependent contraceptive. So I'm showing you results here in mice. On the left, you have AMH levels in the blood through time. And on the right, you have the cumulative number of pups that were born from those females. On the same time, the same animals are on both graphs. You can see that animals in red here that have high AMH levels in their blood, well, after 4 to 6 weeks, they're not able to get any more pups. So they're quite infertile. But animals that have lower level of AMH in blue here, they still conserve a little subfertility with time. So they're not completely infertile, they're just having like smaller litters. And these experiments allowed us to fix a contraceptive threshold. So we're saying that when they are over 0.25 microgram per ml in the blood, those levels of AMH, they are sufficient to make them infertile. So that's like a goal we want to reach, and you'll see it later in our results. So to make sure you're not confused, I need to explain to you why we're using this big name for our contraceptive. So in cats, we'll call it the AV9 FCMIS-V2. AV9 because we're using serotype 9 of AAVs, which was shown in previous studies in other species and also in cats to reach high and durable expression of the transgene. FC because the product is feline domestic cat specific. And to honor our predecessor at MGH that were among the first to purify and clone the AMH gene, but gave it the name Mullerian inhibiting substance at that time, we kept MIS in the name of the product, but keep in mind it's AMH. And this is the second version of the cat contraceptive we've tested. So we first tested this feline-specific contraceptive in mice that lack an immune system to make sure it works in vivo. So for the rest of the presentation, you'll see in black animals that received empty AAV9 particles. So there's no transgene in the AAVs. They are our controls. And in blue and red, those are the treated animals, red being twice the dose of the blue animals. You can see on this graph that the AMH levels are 100 to thousands of times those of controls, and they are over our 0.25 contraceptive threshold. The ovaries of the treated animals are way smaller, and the reason for this is because they have significantly fewer number of those big growing follicles. So we were quite confident it was working in vivo in mice, and then the next step was to go see if it works in cats. So I'm gonna let Lindsey take it off.
So jumping into the cats, I'm going to start just by give you guys an overview of the study design. So this is where all of the research took place for this part of the study. So this is our research facility on grounds at the Cincinnati Zoo. And within our building, we have a whole floor dedicated to cat holding. So for this study, we chose sexually intact, nulliparous domestic cat females. They were 6 to 12 months old at the time of study initiation. They were group housed, as you can see in the picture, with the exception of, for the 1 week following gene therapy treatment, we did house them under BSL-2 housing with single cages. Okay, so again, you'll notice we're keeping the color scheme throughout this. So all of the results you'll see for the high dose cats will be red. Similar treatment scheme. So the females in the high dose received 1 × 10⁻13 viral particles per kilogram. And again, as Philippe mentioned, this is we see dose-dependent effects. So all of this is based on the body size of the females. The low-dose females received half that dose, so 0.5 times 10 to the 13. And then the 3 control females received that empty vector at the same dose as the low dose. To give you an idea of the timeline, each of these circles represents a 6-month time interval. And so before I jump into the results, I just want to give you an overview of the different endpoints and sort of where we looked at them. So we performed 2 breeding trials. They were each 4 months in length, and they ended at year 1 and year 2 post-treatment. We monitored health throughout the study, so 4 years and beyond, also monitoring the levels of the AMH expression. For the first 2 years, basically coinciding with the end of that second breeding trial, we monitored their reproductive hormones, and then more recently monitoring their uterus via ultrasound. Okay, so jumping in. Okay, so of course, health is a big question for us. If we're going to apply this technology to free-roaming cats that aren't going to have regular access to veterinary care, we need to be confident in what some of the side effects may or may not be. So each time, in this case, each of these circles is going to represent a 1-month period. And each time you see a darkened circle, that represents a time point that we performed blood work, so CBC chemistries and physical exams. So 1 month prior to treatment, all of the cats got checkups, made sure everybody was happy and healthy. We repeated that again at time 0, so right before actually treating them with the gene therapy. At this time, they were sedated. We have good cats. They're not this well behaved. So this is Barbara, one of our females, getting her checkup. We sedated them mostly so that we could be confident when giving them the injections that, you know, if there was any difference in results, that we wouldn't think maybe she only got a partial dose, she moved. So that was the impetus for that. And then for the next 7 days following treatment, we followed them pretty intensely to look for any injection site reactions. So of the 9 females, 1 of the females in the high treatment group did have an edema or swelling right at the site of injection. It was moderate in degree, lasting only 2 days. She didn't show any pain. She ambulated normally and it resolved on its own. And we saw no other reactions. So following through the first year of the study, we repeated blood work and physicals every 3 months. And then up until year 3, we monitored them twice a year. And now we are at a point where we're monitoring them annually. And throughout this time period, we have not seen any remarkable changes or off-target effects. Miss Betty is here to remind me to share with the audience that we always adopt our cats after studies that we do at Crew. We've actually started to adopting out these females. And so we are planning to continue watching these females for at least another 5 years. And so what we do is we only adopt these females to people in the Cincinnati area, and then they bring their cats back once a year, physicals, blood work, you know, it's kind of a win-win. They get a free health check on their cat. We get to keep an eye on these cats and get to monitor them long-term while they're getting to be in homes. So of course, feeling more confident on the health side, the next question is, did this gene therapy treatment work? Are we able to actually express AMH in these cats? So again, keeping with the color scheme, every time that you see a shape in black, that is a time point that we drew blood and assessed this. And again, that level Philippe mentioned is noted there, the 0.25 micrograms per mil of serum. That is the theoretical contraceptive threshold. So note that the control females in black, they do express AMH. Again, this is a natural protein made by the ovaries, so we're not surprised to see it. But note, of course, it's well below that threshold, as would be expected. But both the low dose and the high dose females showed a very similar expression pattern. So initially after treatment, you see it go really high, and then it has a steady out, but then a very stable plateau. And notice that we are now out at 52 months. And if you're like me and not great at math, you know, if somebody tells you their kid is 52 months old, how old is that? I don't know. So it's over 4 years now. So over 4 years, these cats have stably expressed this AMH protein, which is pretty exciting and certainly supportive of the notion that this could be lifelong contraception. So the next question is, how does this affect the reproductive hormones? So to answer this, we followed their hormones starting 6 months prior to treatment. Part of that was just to confirm that they all are cycling normally prior to treatment, and then following them out for 2 years after treatment, including those 2 breeding trials I keep alluding to. Now, an important point is, unlike AMH that we can monitor every month, every couple months, and have a really good idea of what's going on. With reproductive hormones, we really need 3 non-consecutive samples every single week. And so you can imagine working in the species like a cat, drawing blood 3 times a week for 2.5 years does not sound, like a very good idea. And so taking a page out of our zoo book, where we regularly have to figure out how can we non-invasively monitor these animals. And so with reproductive hormones, like any hormone, they are in the blood, but they will ultimately be metabolized and excreted. So the question is, what is the route of excretion? And luckily in cats, hormones are excreted. The majority route is in the feces. But recall, our females are all group housed. And so to solve that problem, we had a fun game of each female gets her own special concoction. So this is food coloring. This is non-toxic glitter. So this is made for, humans with children that eat things they shouldn't. So safe to feed, and then rice. And so each female gets her own special little cocktail profile. And then our keepers, literally on the other end of things, get to go through the litter boxes and find these samples. So while it is still a lot of work, what it does is create a really rich data set, and it gives us a good idea of what's going on reproductively. And so the 2 hormones we were most interested in, estradiol here, so that's the hormone in green, and estradiol is, as we like to say, it's the love hormone. When a female has high estrus, she is just that into him. When estrogens are low, a female is not that into him. The other hormone of interest is progesterone, shown here in purple as P4. As the name suggests, progest. So this is the hormone that supports pregnancy. So you saw that nice schematic from Philippe. So after an egg is ovulated in that ovary, that structure will develop, restructure, and form into a corpus luteum and start secreting this hormone. So we can look at progesterone and say, did the female ovulate? And if it's up long enough, then you can say she's pregnant. So before I jump into our hormone results, you're going to get a quick teaser on reproductive physiology. This may sound familiar to anyone who has taken a course on this before, and that cats are really unique in a lot of their strategies. So I would say normal, what most mammals do is with each time they cycle, and that estrogen goes up and down, they ovulate. That's just part of the normal cyclicity. Cats are different. If there's no male around, the cat will not ovulate. She will, in fact, just go down and just immediately go right back into another estrogen peak. And so they have these really short interestrus intervals. Now, if a male is present and there is successful copulation, and in this case, defining successful by the physical act, by that intromission, that will stimulate the female to ovulate. Either she will become pregnant, or she will, even if she doesn't become pregnant, she will still ovulate. She'll just have a short luteal phase. So seen here in this graph, 45 days versus a successful breeding where the eggs are fertilized, she does become pregnant, that luteal phase, that progesterone level will stay up for about 60 days. So after just telling you that, I'm going to immediately tell you that that is sort of an ish. Cats actually do spontaneously ovulate. And so anybody, again, who's had a reproductive physiology course is often surprised because it's never taught that way. However, it does happen. The percentage of females can vary widely depending on the study. Sometimes 1/3 of the females will exhibit this, sometimes upward of 2/3 or almost all. Additionally, the rate. Some females may only spontaneously ovulate 1, maybe 2 times in their entire reproductive lifespan. Other females will regularly ovulate without any copulatory stimulation. Now, anecdotally, there are certain factors. Older females may spontaneously ovulate more. Females that are housed together may spontaneously ovulate with more frequency, or females that have a male presence, such as visual, auditory, olfactory. Now, having said that, we also have documented young, singly housed females without males can also spontaneously ovulate. And so this is really important because it feeds into this disease complex. It's a lot of words, cystic endometrial hyperplasia pyometra complex. And so what happens with females? The first stage is the glands of the uterus will get dilated, and they'll get really cystic. And as time goes on, if this proceeds, that creates the state of inflammatory processes. And that sets the uterus up for an infection. And so if that happens, often the first clinical sign owners will see is a purulent discharge. Anybody's eating lunch or doesn't want to look, there are some graphic pictures, so look away. These are some examples of the surgeries I've done over the year for pyometras. It is a life-threatening condition and can be very serious. Anybody didn't want to see the pictures, you're safe to look again. And so this is all important because progesterone is what plays this primary role. And so we know cats can spontaneously ovulate, and with that comes more progesterone feeding into this disease complex. So now, circling back to the idea of spontaneous ovulation, how can we look at that in our females? One way is using that fecal hormone data and just comparing the average levels of progesterone. So again, with your controls in black, you can appreciate virtually unchanged pre and post treatment, which is to be expected. But in both the low dose in blue and the high dose in red, that average level of progesterone did drop post-treatment. Now, additionally, we can look at this with a little more detail. Circling back to this example profile, hone in on that purple hormone again, we define luteal phases. So look at that line at the bottom, that dotted line. In that case, that is what we consider the basal level of progesterone in this particular female. And anything that is 2 standard deviations above that, we call elevated. And so when we see several consecutive samples in a row with that elevated progesterone, we call that a luteal phase. And you can see it's pretty obvious on the screen. You can appreciate when it goes up, it goes really high and it stays high. So we basically used this technique for all of the females, looked at all of their profiles over that whole 2.5-year time period and calculated the number of luteal phases. And so that's what this graph is showing you on the right. Again, you can appreciate the controls unchanged. Now, the low dose group certainly trended downward to a lower rate, but unfortunately, small sample size, and again, cats, they do what they want. It wasn't statistically significant, but appreciate in that high dose group, there is a significant drop in the rate of spontaneous ovulation, with one of the females going to 0. So taken together, this data demonstrates that treatment with AMH or treatment with our gene therapy to overexpress AMH leads to a reduced rate, it's a lot of words, of spontaneous ovulation. And so that is one way we are monitoring the health of these females. Certainly knowing that information is useful. But there are certainly other techniques we can employ as well. So giving you back this timeline, this is when we monitor the reproductive hormones. Age certainly plays a role with this disease as well. And so while it's not feasible for us to adopt these cats into home and ask the owners to collect poop for us 3 times a week for the next 5 years, what we can do on these assessments is perform ultrasound and look at the uterus directly. And so that's what you're seeing here. Gives us a lot of great information. We can actually see if those cysts are forming. We can also measure the area. And so we began tracking these roughly 2.5 years into the study. And again, following with that color scheme, you can appreciate early on, 2.5 years later, so these cats are roughly 3.5 years old at this point, the areas are very similar. But as time goes on, that control group significantly increased versus our low and high treatment females have remained virtually the same size. Graphic picture number 2, this was one of our control females that we decided to spay due to the pathology we saw on the ultrasound. So again, this was a relatively young female, just still reproductively intact. Okay, the picture is gone for anybody that wanted to look away. So finally, all of this together, the big question is, how does this actually affect fertility? And so we answered that question by performing 2 breeding trials. So these were 4 months in length. So for 8 hours a day, 5 days a week, one of our proven breeder males got to come visit the ladies and stay with them. We actually chose 2 different males. So there was for year 2 had a different male than year 1, just in case there was any sort of, cats have opinions, in case there was any mate choice in there. Unfortunately, unlike humans, with which we have hormones we can detect very early and know if a female is pregnant, this doesn't exist in cats. And so ultrasound remains the gold standard way to detect pregnancy. I just like to show this. So we ultrasounded our females every week throughout these 2 trials. And it's not as easy feat as you think to convince a female to lay on her back and let you put ultrasound gel and alcohol. So lots of true, lots of love. But the nice thing about ultrasound is there's not a lot of false positives out there. It's pretty definitive when you see a pregnancy, as you can see here. So I'm going to take you through the results of the trial. All right, so recall there's 3 females in each of these groups. So the first question was, do these females allow breeding? As expected, all 3 control females did allow breeding with the male. What surprised us was that in each treated group, one of those 3 females also allowed breeding. So then before I jump through this table, one last primer on cat reproduction. Recall that I said they are induced ovulators. So while cats may or may not spontaneously ovulate, that is a lot of individual variability. Absolutely normal cat physiology is every cat that has successful breeding, that physical act of breeding, if that occurs, the female should ovulate. So that's not variable from cat to cat. So as expected, all 3 control females ovulated, but interestingly enough, neither of the control females followed breeding ovulated. And if you look at the row above, you'll notice that female in the low group had 6 separate breeding bouts over these 4 months. Similarly, all 3 control females became pregnant, had a collective number of 10 kittens produced, versus none of the treated females became pregnant. So trial 2 was very similar. It was the same 2 females that allowed breeding, but yet again, no ovulation following copulation for either of those treated females, whereas all 3 control females ovulated after breeding with the male. And yet again, the 3 control females became pregnant, 11 kittens produced in these litters, and none of the treated females were pregnant. So just to sort of summarize this data, treatment with our gene therapy resulted in a decrease in the overall levels of progesterone, a reduction in the rate of spontaneous ovulation, a complete inhibition in coitus-induced ovulation, and a total prevention of pregnancy. And I'll remind the audience again that these levels have remained above that contraceptive threshold for 4+ years now. And certainly data we have with human gene therapy and other species such as the dog suggest this absolutely is reasonable to believe this could be the life of these cats. And so sort of taken all together, I believe this data demonstrates that gene therapy has the potential to replace surgical sterilization as a mainstay way to manage cat reproduction, particularly with these free-roaming cats. And so before I end, I wanted to just take a moment to talk about next steps because the response so far has been overwhelmingly positive. It's been wonderful, all the people we talked to, the excitement, but everybody's ready. Let's go do this tomorrow. Let's go get this done. And so I just thought it would be useful to talk about some of the next steps that have to happen before we're there. So one thing is just defining the final version of this. This was a proof of principle, but this vector was made for 6 cats. And now we have to think about millions of cats. And so making it optimal, how do we optimize this to maximize the amount of AMH that's produced, to maximize that vector's ability to get to the muscle cell via the route of how we're giving it, and then similarly, make it easy to produce. And I say that because another important step is finding a way to scale up the manufacturing platform, right? Good quality control, but scale this up. Not my area of expertise, but I can tell you that how to produce these vectors, how to produce this gene therapy product is not easy. There are a lot of steps, and it is, again, why we're very grateful that we work under the umbrella of the Michelson Fan Animal Foundation, because part of their team has this expertise. And so they are the ones that can actually bridge this next step and come up with this platform. Similarly, regulatory approval, just as drugs that are produced for humans need approval, so will this. And it also falls under the US Food and Drug Administration here in the US. They have a whole arm dedicated to veterinary medicine. Again, I will say that is not my forte. And again, be grateful to work with Michelson, who again has a team of experts who's familiar with the regulatory process, knows how to go through these steps, knows how to negotiate with them, how to design the studies that the FDA will want to see to ensure that this is a safe and effective product. And it's not just the US, of course. This is a worldwide problem. Hopefully once FDA approval, if and when, FDA, I'm not saying you're going to approve it, so don't quote me, but if and when this approval occurs, hopefully that can make the international process easier, but that will be country by country. And then I'm just going to end by saying public education will be an important key of this as well. Gene therapy sounds scary to a lot of people. And now I'm teaming that with, sterilizing, stopping reproduction. I'm pretty sure they've made a couple movies about that before. So, I think we have to be really cognizant of that and really do a good campaign so the public understands. To give you an example, this was a study. This was a group doing clinical trials for gene therapy in humans to not just treat, to cure HIV. And all of these participants were all currently infected with HIV and were given the option to participate. And this was their comments, scary, frightening, I'm afraid. One participant asked if this might make a sixth finger grow. And that's not, without education, that's where it goes. So there's a lot of steps on several fronts, long road ahead. But we're on the road now. We are on that road. And so this breakthrough is such an important first step to getting there. So I believe that is it. I just wanted to show the slide. I'll give Philippe the credit. He put this together and I love it. But you can see all of our teams from each of our institutions, of course, all together under the umbrella of Michelson, and then our other funding sources. Up in the corner, hopefully my face is in the way, it was on me, but there's a QR code that actually links to our paper, so you don't have to go find it. You just click it and it'll take you right there if you want to read more. But that is it. I'm wondering if I should stop sharing the screen so everybody can be up again. Maybe that's what I'll do.
I would actually leave it up so that if
I
they
would
want
say
to,
leave it up.
yeah.
Okay.
So if they want to get to the QR code and want to
Perfect.
download that paper while we're all doing the Q&A. Lindsey.
Stephanie, can I take 1 minute before we
Sure.
launch into the questions?
Sure.
I just want to introduce that we have Thomas Conlon on our panel today. And he is Chief Scientific Officer and Scientific Advisory Board Chair of the Michelson Found Animals Foundation. So I welcome him as well. And just maybe giving people an extra minute to type in their questions there. And I'll leave it to Stephanie now.
Thank you for joining us, Thomas.
Thank you.
Okay. So first of all, outstanding presentation. This is all really, really exciting. I'm going to start with a very simple question. So everyone was interested to know where the injection site location was.
No, so it's an intramuscular injection just into like the quadriceps. So like the back leg of the cat, kind of that meaty front part.
Okay, and then I'll let you and Philippe decide which one is going to take which question once I read it to you. Okay, so first question or second question, would it be feasible to perhaps use microspheres for the slow release of AMH during a prolonged period of time, simulating the 40-day injection as a cheaper alternative of AMH release?
Yeah, so microsphere, they're a way to like slowly release a drug, like you would put that under the skin or something like this. And like osmotic pumps are doing the same thing. So it's kind of a device or a technique to slowly release a drug. I'm not an expert in these, but I don't think they would last like multiple years. We're more talking about days or maybe a month. So it wouldn't be like ideal for the goal we have in mind in that case.
Okay. Next question. A comment. We would love to try your trial on the Mountain Ute tribal lands. It is a semi-closed population and is not regulated by USDA as it is a sovereign nation. It would be a perfect place to test it. So just food for thought. Let's see here. Any idea about cost projections? I'm not sure if that's something that you all can really speak to at this juncture, but any thoughts on that, or is that sort of the next step process you were talking about, Lindsey?
I think that's a great question to kick to Thomas.
Yeah, thank you. And a great question. Our goal is to target a similar price to a surgical spay or neuter by providing it as an alternative to surgery. It's as Lindsey mentioned, the manufacturing of this has a long way to go, especially in the quantities we're going to need. But because we're going to be manufacturing so much, that'll bring the cost per dose down. But again, our goal is to make it equivalent or even less than surgery.
Okay. Here's an interesting question. In a little bit, I had this question kind of rolling around in my head as well. I think you did you examine any behavioral changes in the female cats treated? You said unremarkable with respect to impacts. I think what they're trying to get to is for what would be a free roaming cat population, if the normal territorial behavior is maintained, if it stays intact, unlike surgical sterilization, it could be a great advantage for population management, as those females would serve as placeholders in their environments and potentially prevent immigration in from other animal populations.
No, I think it's a great question. This comes up a lot in various forms of what roles do the sex hormones have in territory? And it certainly does make sense. Often it's males that are territorial, but certainly just the need for resources, I think, drives that as well. I think it's hard to assess those behaviors in our very spoiled cats who don't have to resource guard. They're all living together in a room. So we certainly, I think that's hard to comment on. Would that remain? I would suspect it would, from a sense of needing resources, but it's a great point, and I don't know that we'll know until we're able to sort of do some field studies and see, does that affect differences?
Okay. And here's another question that I was thinking. I'm glad everybody is following my track of thought here too. Did the treated females exhibit vocalizations related to reproductive cycles? So during the study, and then are people that are now caring for them talking about that at all? Are they required to kind of monitor behavior and then write what happens? I'm curious to know that too.
It's a wonderful question. So it's hard for us to monitor estrus in the colony. A lot of people will know there's a classic like lordosis state, where you kind of touch the cat in the back and you do lordosis. Our cats that are involved in any kind of reproductive study that we're testing this learn that that's a good way to get behavior. So lordosis is not a good endpoint for us. They don't act, this is very non-scientific. They don't act like intact females. Like, we have rooms of still intact females in our colony, and they're monsters. They're very different cats. With regards to their behavior in the home, that is something we're very interested in. We went back and forth on the best way to ask that question. We don't want to lead these owners into, are you looking, do you see estrus? Does she, yeah, we wanted, are they normal cats? And so we asked very open-ended questions. Of course, they are aware of the study their cat was involved in. But so far we basically asked broad, any behavioral issues, describe your cat, and there's been no reports of late night meowing or every Tom in the neighborhood showing up or anything like that. So I think the more cats that get treated and get adopted, we can answer that better. But our very small sample size says they act more like spayed cats than intact cats on that front.
Okay, I think you all knew this was coming because we're the Bot-Stieber Institute for Wildlife Fertility Control. Could you use this on free roaming wildlife? If so, will it need to be approved by EPA in the US and not the FDA? And actually, my question also is, if you're using this with free roaming cats, and that's really your intended target, does it have to go through FDA? That might be a loaded question. If you don't feel comfortable answering it, that's fine. But I'm really wondering that, because for other free roaming wildlife, these types of agents are registered through the EPA. And so, yeah, just curious.
Thomas.
Yeah. For it to be, we plan on it being a product available to veterinarians and the TNR field for cats. And therefore, because it would be a drug used by veterinarians, it would need to be approved by the FDA. We haven't discussed really other applications outside of cats and dogs with the FDA or other regulatory agencies to determine where the approval would need to come from, for such as horses, for example. And I'll let Philippe further talk about whether the product that we're talking about today could be used in other species.
Yeah, so the product we presented is feline specific, so it would only work in domestic cats. So you would need a species specific product for each animal you would need to try it on. So you would need to do this preliminary results first, and then like go higher. So this product specifically only for cats, but it would be feasible in other species. As I showed you, AMH signaling is well conserved in many other species. But yeah, it's the idea.
So basically the concept, it would be the same, but you would have to create an agent that is species specific depending on what your target species is, correct?
Yeah, correct. The approach would be the same, the strategy would be the same, but you would need to make the product species specific. And also one thing to consider is we give the product on a body weight dosage. So for like bigger species like wild horses or deers, it would be difficult to have a product like affordable, I would feel like, because you would need such high volumes of this product. But I don't know what the future will bring us in the AAV technology.
With wild horses, animals that live a long time and are hard to access, and the cost benefit of treating them once and then not having to treat them again, depending on the context, that actually may be more cost beneficial than what is happening right now. So to your point, it really is species and context specific. Because compared to a cat, it may cost a lot of money, but compared to how the animals are being managed now, depending on what country is doing it, they may see this long term as a wiser way to do it. So how was the sample size determined to assess quality and safety, or was that not really the point of it for this proof of concept trial?
I can't say we did like power analyses of like, if we do 20 cats, we'll see this deviation in CVCs or something like that. This really was a proof of principle, just to see if it expresses and it works. There'll be much larger studies still underway to actually assess safety. And again, a lot of that depends on conversations with the FDA of what they want to see done. But certainly hundreds of cats, I would anticipate, for the larger safety and efficacy studies.
And then go ahead, Philippe.
If I can add, since we were looking at complete infertility, it's a black or white endpoint. So we didn't need like hundreds of animals for that first study, and we got the results we were looking for.
And then any sedations for injections, are those going to be recommended in the future, do you think? Or
I think that's a question we keep having too, is, great, this is field specific, field friendly. I don't think so. I think once we have a much better idea of what the dose is that we need, it's a simple IM injection. Of course, it does depend whether we're talking about, a free roaming cat versus a truly feral cat, the type of cage, these questions of, will you be able to successfully give the full dose? But I think that will, if that's the only injection you have to give that cat at that time, I think it'd be reasonable. Most of the cats you wouldn't need sedation for. Again, for us, it was more of a making sure there's no variability in the results, because one of our girls wiggled while we were giving her the shot, just to be confident in that the full dose was given. But it's not, I guess I can't say it's not painful. I don't have any reason to believe that it would be. It's not a lot of volume, and with better, with optimizing things, the volume will probably go down. It was less than 1 ml per cat per injection site. So most vaccines are 0.5 ml to 1 ml.
And this is a kind of a group of questions. This person wants to know, would TNR surgery in this context still be encouraged for males? Would vaccines be included with this gene injection? So would you use this as an opportunity to not only use this agent to essentially sterilize the queens, but would you also be using it as an opportunity for administering vaccines And then ear tipping, they want to know if ear tipping would be done, so you know which animals you've treated and which ones you haven't. And then they also want to make sure that they understood that this form of gene therapy prevented pyometria as well as pregnancy. Is that the case?
So let's see.
Sorry,
So
I told you it was a group of questions. So,
and anybody else that wants to jump in on these, jump in with me. For sure, neutering is the only option in males. We would have liked to have had it work in both. I believe Philippe mentioned this, but part of AMH's role actually prenatally is driving towards male. So males naturally express a higher level of this hormone through puberty. So unfortunately, it doesn't work in males. But if you're going to pick a sex to be able to non-surgically sterilize, I would argue female's the way to go, because cat neuters are much easier to perform than a cat spay. So neutering for sure in the males still. I think it depends on the TNR group. Most, I do believe, have a policy of vaccinating for the respiratory diseases and rabies. So absolutely, that would be something that would be done at the same time. But I don't know that would be past, that sort of end user choice of those groups. But I would hope that would be part of the plan, because you want these cats that are still out in the population to be vaccinated. I'm trying to remember. The pyometria question, I can't say 100% these cats won't develop that. It's too early. That's what a lot of these larger studies are needed for. It was a concern of mine early on, but I was very happy with the data we have so far, albeit a small data set that certainly suggestive that with the reduced spontaneous ovulation, the lack of induced, they're not seeing nearly as much progesterone. These cats are now 5 years old. You saw what the control uteruses are starting to look like. But that's another question. We need a lot more cats treated and a lot more years to say what is the relative risk of pyometria in this case. I will feel confident saying it's going to be reduced versus them being intact, but I can't say it's 0 versus a surgical spay.
and I think Dr. Conlon may
need
have already answered this question, but who would be authorized to perform this injection when you are at the point, if you get there, of having a scaled product? Would this be considered a procedure that is considered a veterinary procedure? Or is this something that could be used by somebody that's working under the supervision of a veterinarian? What is this may not be worked out yet, but what is the thought process behind that?
Right. We would be seeking approval to have it administered by under veterinary care, under a veterinarian's care or observation.
Okay. And then from a practical point of view, from population management, would more than one dose be safe for cats? What if it was accidentally given to a male cat, for instance?
So male cats already have quite high AMH levels, like naturally. So I wouldn't bother that much of adverse effects in males. And we have preliminary results showing that we injected males, but it didn't prevent them from being fertile. But to answer your question on giving twice the dose to an animal, we would use the same marking technique of spaying females in the wild, ear tipping or stuff like that. But we will do a safety evaluation to make sure that there is a good safety zone for this product.
Okay. And I think we have time for just one more question. And this is an interesting one. So what are the chances of engineering this product so that it could be delivered as a bait to be consumed by feral cats? Since the product is cat specific, presumably it would not harm other species. Would the product, if it could be manufactured as a bait for oral delivery, have any adverse impacts on male cats, which I think, Philippe, you just addressed? But is that even remotely feasible?
Regarding using it in bait, viruses tend not to stay intact outside of their target host and at temperatures, in particular temperatures, normal body temperatures. Soon the virus starts degrading. So if it was used in bait in warm areas or even those above refrigerator temperature, it wouldn't survive longer than, a couple hours perhaps.
Okay. Great. Well, Monique, I'm going to hand it over to you. Thank you all. And for those that since we ran out of time, for those that did not get their question answered, we have somebody that's going to relay these questions to our speakers, and they've graciously agreed to answer them. So they will be getting back to you at their earliest convenience. So Monique, I'll hand it over to you.
Yeah, and I would just add that if you develop questions later, that you can email them to the Bot-Stieber Institute, and we'll see that they're forwarded to our presenters, our panelists. And with that, yeah, that was a fabulous presentation, and we had a lot of interest in this particular webinar. So I'd like to thank everyone, Lindsey, Philippe, and Thomas. Thank you very much for being our panelists today and bringing this to our audience.
It was a pleasure. Thanks for having us.
Okay, and with that, we conclude our webinar. Thanks everyone for attending.